Hello, this is Tommy. Welcome back to Genomics. In today's video, I'm going to show you if you master the 6 types of plots, you can make 90% of the figures in any genomics paper. I have talked about this for many times. If you want to improve your bioinformatics skills, or if you want to gain practical bioinformatics skills, try to reproduce a figure from the genomics paper is the best way to do it. And in the end, actually, I'm going to prove you that the 6 type of plots actually are present in a genomics paper. I'm going to show you with a real example, and let's actually go through that together. Now let's dive in. So the first type is called the bar plot. So bar plot is very common in any genomics paper, or actually in any paper, not necessarily genomics paper. So the y-axis is just the number or amount, right? So you can use that to compare the number among different groups. For example, in here, you have the y-axis, maybe just the gene expression of a particular gene. And you have maybe 3 different genotypes, A, B, and C, and 3 different time points, time point 1, 2, and 3. And you can use the bar plot to look at the differences across different time points for a particular gene or protein. Of course, you can rotate it just 90 degree and you still get the group bar plots. And this is the stacked bar plots. This is a very commonly used to look at the composition of cell types in each sample, for example, in a single cell RNA-seq paper. So the next plot type is the scatter plot. So scatter plot is very frequently used to look at the relationship of 2 different variables, x and y. So in this case, you have x-axis here and y-axis here. And for example, you may have 2 genes, gene A and gene B, and each point here could be one sample or could be one single cell. So you're looking at whether those 2 genes are correlated, for example, across the samples. The bubble chart, essentially, you are actually mapping a third variable, for example, to the size of the dots. For example, it could be percentage of cells express that gene, right? And this slope graph this is also just the scatter plot, because you have 2 points, but they are actually connected with a line. This is very frequently used to look at the value, for example, a gene before and after treatment. Then you want to look at the treatment effects of that for that gene. So, and in this case, you want to say, okay, maybe there's an increase, there's a trend for increasing that gene after treatment. Next will be the line graph. So line graph, this is essentially just use the line to connect the dots, then it became the line graph. It can be used to look at the trend of a certain variable, maybe, for example, the tumor size. Then you have different time points here, then you measure the tumor size, and as the time goes on, then the tumor becomes bigger and bigger, for example. And you can also have this smooth line graph, so if you just smooth it, then you get a smooth line graph. Okay, the next type is a histogram. Histogram is used to visualize the distribution of a single variable. So in here, the x-axis here is just the different bins, right? You can bin the values into different bins, and then you just count how many samples fall into that bin. And so essentially, y-axis is the number of samples fall into that bin. And if you make the bin size infinitely small, it actually gives you this density plot, right? So it actually looks very similar in terms of the shape, because if you just make the beam infinitely smaller, okay? And the next type is the box plot. So box plot probably is the most common plots in any genomics paper. This is used to compare distributions across different conditions or groups or categories. And you show you, here is the median, then you have the 75 and 25% quartile. And for example, you can look at the distribution of a particular gene. So in 3 different categories, like adjacent normal, Maybe this is the tumor, and this is the metastasis tumor samples. And violin plots are also used to look at the distributions and compare the distributions of different categories, but it add another layer of information, which is the density of the data point. So the width of that violin tells you, okay, how dense, how many more actually samples go into that region. Okay, for example, then you know here it's wider, so you know there are a lot more samples here. And the strip charts or just dot plots here, it's just the underlying actually data points for those plots. So each dots here will be just one single data point. And in practice, like people usually lay those data points onto those box plots and the violin plots. So you have both distributions, but also the raw underlying value. And for all of those previous plots, you can easily make them using ggplot2. So master this basic skills, how to use ggplot2, and you can make all those type of plots. And the last type of plot is called heatmap. So heatmap is of no mystery. This is just use the color to represent the values. So in here, for example, we have x-axis to y-axis here. So we have rows, for example, genes, maybe columns, samples here, and the numeric value will be just the expression level of a particular gene. And then you can use to actually look for patterns, and you can do the clustering for the rows and for the columns, and you can show the dendrogram. And I like to use actually complex heat map of this bioconductor package to make heat map, because it is very versatile and really, I would say, flexible. You can make very complicated heat maps. For example, you can add those bars on the rows and columns and the rows here. Okay, so I have shown you like the 6 type of plots, and I'm going to prove you that in a genomics paper, essentially 90% of the figures, they are those 6 different types of plots. Okay, this is the paper I'm going to show you, genome-wide association between YAP, TED, and AP1 that enhances drive oncogenic growth. Okay, this paper, they performed a lot of chip-seq analysis, and for example, in figure 1 here, okay, actually figure 1A and B, those are Venn diagram, which I didn't talk about in my slides, so that's why I said 90% of the plots. But if you can include actually a Venn diagram as well. If you know how to use R Python, this is actually very easy to use. There are packages made for that. And for figure 1C, this is just a line plot, right? And figure 1D here, you have the x-axis and the y-axis and the y-axis here, and then you have points here. This is a bar plot, stacked bar plot. So G here is the IGV genome browser view of the chip-seq signal for different factors here. So people usually just take a screenshot from those IGV, for example, genome browser. But if you analyze the figure carefully, those are just histograms, right? So x-axis are the genomic locations, genomic coordinates, and then the y-axis is just the signal, right, of those chip-seq signals. And if you actually zoom in the IGV genome browser, if you zoom in, and you will see actually those are just histograms, okay? So you can make those histograms actually using just R or Python. And H, this is the pie chart, which I didn't talk about. And figure I here, it's actually a very common figure for chip-seq. And if you analyze it carefully, this is just a heat map. So you have each rows are a region or a region actually. Then each column here just a being around the summit of the peak. Okay, so for example, each row here, like for example, one row here could be just one region like this, and this center is just the center of the summit, and then you have like maybe 5 kb or 5 kb upstream and 5 kb downstream. Then you have maybe you use like 100 base pair as one bin, and then you have like 50 bins upstream and 50 bins actually downstream, right? And then just count how many reads in those bins and then represent them as a heat map. And J, this is just a line plot. K, this is just a bar plot, right? And again, let's look at figure 2, pie chart again. And this is a heat map. It's a line plot, bar plot, line plot, bar plot, line plot. This is like the chart, the chart and plot, right? We essentially look at distributions of different groups, and then this is line plot, heat map, pie chart, genome browser view, histogram. This is the bar plot. Let's look at the last figure, heat map, and there's a bar plot, heat map, bar plot. Then this is the stripped chart. Let's see. There's a line plot. Okay, that's all the figures, right? About 90% of figures, they are actually of those 6 types that I have shown you. And in fact, I'm going to actually make an end-to-end tutorial how to reproduce this figure 1 in this paper. And from downloading the raw FASTQ files for the chipsake, process them, and make those figures within R. So make sure actually you subscribe to the channel. I'm going to actually make the tutorial during the holidays, and then please stay tuned for my next video. So that's it for today, and happy learning, and see you next time.